T cells in the thymus undergo a series of gene segment rearrangements similar to those of B cells. Developing T cells face a similar challenge to developing B cells. They must assemble a functional gene for each T cell receptor chain, while at the same time ensuring that each T cell expresses receptors of only one specificity. Not surprisingly, T cells follow an almost identical strategy to B cells, in that the receptor is assembled in stages, with each stage being checked for correct assembly. Moreover, as in B cells, a productively rearranged gene is expressed as soon as it is made, and the products are assembled into a receptor complex, in this case either a pre-T cell receptor or a bona fide T cell receptor. Expression of this receptor is instrumental in promoting further development, which ultimately results in shutting down further rearrangement at the locus that has been active. Despite the similarities with B cell development, the control of antigen receptor assembly during development is more complicated for T cells because there are two different kinds of T cells that could be generated from an undifferentiated precursor: the alpha-beta T cells or the gamma-delta T cells. These two types are distinguished by the different genetic loci that are used to make their T cell receptors. Thus, the T cell developmental program must control to which of the two lineages a precursor commits, and must also ensure that a fully developed T cell only expresses receptor components of one or the other lineage. Another key difference between B and T cells is that the final assembly of an immunoglobulin leads to cessation of gene rearrangement and initiates the further differentiation of B cells, whereas in the case of T cells, rearrangement of the V alpha gene segments continues unless there is signaling to positively select the receptor. And that's T cells in the thymus undergo a series of gene segment rearrangements similar to those of B cells. Have a great day.